What do Bahrain and the Indian Ocean island nation of Seychelles have in common? According to the Wall Street Journal, they have both relied predominantly on the China-produced Sinopharm COVID-19 vaccine, and both have also experienced recent spikes in infections, despite large percentages of their populations being fully vaccinated. The picture, though, is a bit more complicated than it seems. Here's what you need to know. Doubts have arisen around the efficacy of China's Sinopharm vaccine after Bahrain began recommending over 50s with comorbidities get a booster of Pfizer-BioNTech 6 months after their second Sinopharm shot, according to the Wall Street Journal. The Sinopharm vaccine works by teaching our immune systems how to attack the coronavirus using inactivated viruses which can't replicate, according to the New York Times. Once injected into a patient, inactivated virus particles are swallowed by antigen-presenting cells that break them up and present fragments of them on their surfaces. From there, helper T cells can attach to the fragments and send signals to activate other parts of the immune system, including B cells and cytotoxic T cells, according to Encyclopaedia Britannica. Cytotoxic T cells attach to infected cells and subsequently program them to die, according to the journal Immunobiology. B cells create antibodies that attach to viruses to prevent them from invading cells, according to the New York Times. The idea behind the Sinopharm vaccine is that the body's immune system remembers this response using memory B cells. It is then ready to help reproduce the same specific antibodies if it comes into contact with a functioning coronavirus, according to the New York Times. The Washington Post reports Bahrain may have been prompted to recommend Pfizer-BioNTech booster shots after the country saw its worst wave of cases since the pandemic began in recent weeks. This came despite having vaccinated almost half of its population, mostly with the Sinopharm vaccine. Bahrain's Undersecretary of Health, however, pointed out to the Wall Street Journal that despite the recommendation, more than 90% of people hospitalized in his country's current COVID-19 wave had not been vaccinated and insisted that Sinopharm offered a high degree of protection. In Seychelles, it is a similar story. Despite 63% of its population being fully vaccinated and 57% of those receiving Sinopharm doses, in May, average cases rose to 400 new cases daily, up from around 50 in April, according to the BBC. However, similar to Bahrain, of those who required admission to a hospital, 80% hadn't been vaccinated. The country's health commissioner, Jude Gideon, underlined the meaning of that statistic in an interview with local newspaper Seychelles Nation, saying, "All the approved vaccines are effective at preventing severe illness and death from COVID-19. We know from national experience and report that they are less effective at preventing transmission. So at least one of the effects we may be watching here is that the Sinopharm vaccine prevents serious illness developing, but doesn't prevent the coronavirus from spreading." However, there are still more explicit doubts expressed about the vaccine's efficacy. According to the BBC, the head of China's Center for Disease Control and Prevention, George Gao, appeared to address the issue when he said, "We will solve the problem that current vaccines don't have very high protection rates," in a press conference reported by the Associated Press in April. What's more, according to the Wall Street Journal, an unpublished real-world study of Sinopharm in Serbia found that 29% of 150 participants had zero antibodies against the virus 3 months after they received the first 2 shots. Olgica Jerkovic-Jakovic, the doctor who headed the study at the University of Belgrade, told the Wall Street Journal directly, "The Sinopharm vaccine is not immunogenic enough, and it appears that its impact is especially low on elderly recipients." For its part, the WHO's assessment of the vaccine, based on multi-country trial data from tens of thousands of people, was that it was 79% effective against symptomatic SARS-CoV-2 infection 14 or more days after the second dose, and vaccine efficacy against hospitalization was 79%. However, that briefing partially acknowledged one widespread claim against Sinopharm that not enough trial data has been released about it, saying that safety data was limited for people above 60 years of age. Here's how other vaccines work. The U.S. looks set to have a third COVID vaccine approved by the government and ready to be distributed within the month. But this vaccine works differently than the Pfizer and Moderna mRNA vaccines that are already being used in America and is effective with only a single dose. Here is what you need to know. Johnson & Johnson's COVID vaccine uses a genetically modified common cold virus, adenovirus 26, altered with blueprints for the coronavirus's spike protein. This same approach is used by the Oxford-AstraZeneca vaccine. The coronavirus's outer coating is covered in spike proteins, which gives the virus its crown-like appearance. The spike protein possesses receptor binding domains, or RBDs, that the virus uses to pry open receptors before penetrating the cellular membrane. The Johnson & Johnson vaccine carries genetic material from the code for the spike protein that the coronavirus uses to enter a human cell. The spike protein gene is cut from the coronavirus and inserted into a vector, a virus that is weakened so that it cannot replicate inside the human body after injection. The EU this summer approved Johnson & Johnson's Ebola vaccine, which uses the same technology. The adenovirus vector also does not alter the human genome itself, as it does not carry the enzymes needed to edit DNA. Once the vector delivers its genetic payload to a cell, it causes the cell to produce spike proteins. These spike proteins are harmless on their own, but they should trigger the body to mount an immune response. This response produces antibodies and memory cells that will recognize SARS-CoV-2, the actual virus that causes COVID-19. While Johnson & Johnson's coronavirus vaccine uses the same adenovirus mechanism as the Oxford-AstraZeneca vaccine, there are two key differences. First, the Johnson & Johnson vaccine only requires one dose. The Oxford-AstraZeneca vaccine requires a second dose given 4 to 12 weeks after the first. Second, the Oxford-AstraZeneca vaccine was 90% effective in preventing COVID-19 in clinical trials. Johnson & Johnson has yet to release full data from its trial, but according to its press release, the vaccine was 66% effective in preventing moderate disease 28 days after injection. However, the company said in a press release that it was 85% effective at preventing severe COVID, and no one died or was hospitalized within 28 days of getting their shot. By comparison, Pfizer and Moderna's vaccines, which uses a relatively new technology that relies on a strand of genetic code called mRNA or messenger RNA, showed more than 90% efficacy from the Phase 3 trials. Last month, Russia became the first country in the world to approve a vaccine for COVID-19. Now, the first batch of the vaccine is ready for public distribution. The vaccine is called Sputnik V in homage to the world's first satellite launched by the Soviet Union. Here's how the vaccine works. The outer coating of SARS-CoV-2, the virus that causes COVID-19, is covered in spike proteins that give the virus its crown-like appearance. The spike protein possesses receptor binding domains, or RBDs, that the coronavirus uses to pry open receptors before penetrating the cellular membrane. Sputnik V employs a gene with the code for the spike protein that the coronavirus uses to enter a human cell. The spike protein gene is cut from the coronavirus and inserted into a vector, a virus that is weakened so that it cannot grow inside the human body after injection. For its vectors, Sputnik V uses adenoviruses that cause the common cold. Two different adenoviruses, RAD26 and RAD5, are employed for the first and second doses to boost the effectiveness of the vaccine. The genes from these adenoviruses that cause infection are removed, while the gene with the code of the spike protein is inserted. Adenoviruses are popular as vectors because they are easy to engineer. They are deemed safe because they have been with humans for thousands of years. The modified viruses should mimic a real coronavirus infection and teach the immune system to recognize COVID-19. Although they do not cause infection, the modified viruses should still trigger the body to produce antibodies that would neutralize the coronavirus. The vaccine is being developed by the Gamaleya Research Institute of Epidemiology and Microbiology in Moscow. Russia's health ministry said on Tuesday, September 8th, that the first batch of the vaccine passed all necessary quality tests, and a countrywide COVID-19 vaccination campaign will begin next month. However, serious questions remain. The vaccine has not even started Phase 3 trials, and if something goes wrong with Sputnik V, it could seriously harm public perception about a COVID-19 vaccine. Quoted in The Lancet, Dean of the Brown University School of Public Health said, "We have no idea whether this vaccine is safe or whether it works. I do not think it makes sense. The difference between doing things correctly and not doing things correctly is a matter of a few months. It seems like a very small gain, and the middle of a pandemic is not the time to be cutting corners." Still, the vaccine elicited a strong immune response in all 76 patients tested in its Phase 1 and Phase 2 trials. We won't know for sure until a Phase 3 trial has been conducted and Russia has made the results available. Would you get the Russian vaccine now if you had the chance? Or would you wait for this vaccine from Oxford University, which is currently in Phase 3 testing on 30,000 patients in the US, the UK, Brazil, and South Africa? The University of Oxford and AstraZeneca say their vaccine is 90% effective in preventing COVID-19 in clinical trials. The vaccine works in a different way than those from Pfizer and Moderna, and it may be easier to distribute. Here is how it works. The COVID-19 vaccine made by AstraZeneca and the University of Oxford uses a genetically modified common cold virus from chimpanzees, altered with blueprints for the coronavirus's spike protein. The coronavirus's outer coating is covered in spike proteins, which give the virus its crown-like appearance. The spike protein possesses receptor-binding domains, or RBDs, that the virus uses to pry open receptors before penetrating the cellular membrane. The AstraZeneca-Oxford vaccine carries a gene with the code for the spike protein that the coronavirus uses to enter a human cell. The spike protein gene is cut from the coronavirus and inserted into a vector, a virus that is weakened so that it cannot grow inside the human body after injection. The AstraZeneca-Oxford vaccine's chimpanzee adenovirus vector was previously used to make an Ebola vaccine. The chimpanzee adenovirus is genetically modified so that it cannot infect people, while the gene with the code of the spike protein is inserted. Once the vector delivers this genetic code to a cell, it causes the cell to produce spike proteins. These spike proteins are harmless on their own, but they could trigger the body to mount an immune response. This response produces antibodies and memory cells that will recognize SARS-CoV-2, the actual virus that causes COVID-19. Unlike Pfizer and Moderna's vaccines, the AstraZeneca-Oxford vaccine does not need to be frozen, which would make it much easier to distribute. And the AstraZeneca-Oxford vaccine involves a more traditional method by using a harmless virus as a vector. Pfizer and Moderna's vaccines, if approved, would be the first vaccines ever approved by the FDA to use messenger RNA technology. Pfizer and BioNTech announced on Monday, November 9th, that their coronavirus vaccine is more than 90% effective in preventing COVID-19 in those who have not previously been infected with the virus. Here is how the vaccine works. An mRNA vaccine is based on the genetic sequence of the novel coronavirus and not the actual virus. mRNA is short for messenger RNA. Pfizer and BioNTech's SARS-CoV-2 vaccine uses the genetic blueprint for the spike protein that coats the virus, according to information published on Pfizer's website. The spike protein is used to synthesize an mRNA sequence. This is packaged into a lipid nanoparticle carrier. Lipid nanoparticles protect the mRNA from degradation inside the body and facilitate its delivery to the targeted cells. The lipid nanoparticle carrier transfers the mRNA and its copies of genetic instructions to the cytoplasm of a cell, where the ribosomes are found. Once inside the cell, the mRNA instructs the ribosomes to produce the coronavirus's spike protein. By itself, the spike protein is harmless. The spike protein is then displayed on the surface of the cell. This simulates a coronavirus infection, thus triggering the body to mount an immune system response. Reuters reports that Pfizer and BioNTech said that they have found no serious safety concerns and expect to seek U.S. emergency use authorization this month. Phase 3 clinical trials enrolled 43,000 participants and it reported only 94 lab-confirmed COVID-19 infections. So when might this vaccine be available at a medical facility near you? Pfizer and BioNTech said if granted authorization, they could produce up to 50 million doses in 2020. Each vaccine requires 2 doses. Then they could produce 1.3 billion doses in 2021. Of course, someone will have to pay for this. In July, the two companies reached an agreement with the U.S. to supply 100 million doses for $2 billion as part of the Trump administration's Operation Warp Speed. Oh, and the vaccine has to be stored at temperatures of minus 94 degrees Fahrenheit. But as Anthony Fauci told CNN, the bottom line is, as a vaccine is more than 90% effective, which is extraordinary.
Conventional pesticides are a double-edged sword that scientists are now seeking to replace with more natural RNA-based plant vaccines. Without pesticides, roughly 70% of the world's crops would be lost to pests, but such chemicals are toxic, killing insects but also affecting the rest of the environment. A new approach developed in Finland and France involves directly spraying plant leaves with an RNA-based vaccine that inoculates against specific pests or pathogens. The vaccine triggers a process called RNA interference, which prevents invading RNA strands from carrying out their functions, thus causing the pest to die. RNA molecules in the vaccine do not negatively affect the host plant. The RNA also has the added benefit of being biodegradable because it breaks down quickly. Instead of chemical synthesis, scientists use a bacteria-eating virus called a bacteriophage to help generate the RNA. But with no relevant legislation in existence to govern its use, it's hard to say when the vaccine will be available commercially.
For more news animations and explainers, hit the subscribe and bell button to join the TomoNews family. Thanks for watching.